Hello, everybody, and welcome to this week's biology lab. Because of the snow day, we're not meeting in person, but because we're equipped to do so much online, then we're not canceling lab necessarily. You just have an online assignment that's due. I'm doing a voiceover this time because unfortunately I got sick over the weekend, and I figure you should just be exposed to my sick voice, not my sick self. So, standing in, of course, we have Ravioli, who is sporting her winter wear. I think she looks just precious in it, but she's a little insecure about it, so you'll have to tell her what you think. We also have Leo, who's her older brother, and he also has the same outfit. We'll talk a little bit about this outfit later and who wore it best. But this is science class, not cat class. So, this week, you're going to virtually carry out an experimental procedure, collect and display data. You'll determine how enzyme activity is affected by several conditions, and then you'll write up your first lab summary, which will include a graph and will include a citation as well, and upload it to Canvas, as well as your Flipgrid video for the week. Typically, we do not do Flipgrid videos for labs that we meet in person. And technically, since you are Group A, then we will be meeting in person this week, but on the calendar, but we are physically not meeting, obviously. So, we're doing a Flipgrid video. All right, our investigation. Why do some fruits and vegetables turn brown faster than others? So hopefully you've read the background. If you haven't yet, I would go ahead and pause right now, go read the background, and come back because there's quite a few things that I'm going to be mentioning that assume that you have read the background. So here's what we would have been using in class. Our main tool is the colorimeter, and that would help us determine our dependent variable, how much light is actually shining through something. We have little cuvettes that the liquid will go into that goes into the colorimeter, and then tubes and droppers that you would have been using. The idea is that while you're setting up, I would have been preparing this potato solution for you guys as you're getting ready for the lab. So this potato solution essentially helps us see the browning of the potato. So as you all have seen probably, whenever you cut up fruits and veggies, a lot of them will turn brown after sitting out for a while. So you cut up an apple. Some of you may have had a mom or dad that would put your apples in the fridge if they're cut open, or maybe would pour some lemon juice on the apple slices to keep them from going brown, maybe if it was a school lunch or something like that. So we'll talk about why some of these tactics can help with preventing browning in different fruits and vegetables. Our vegetable of choice is a potato here. So this browning is caused by a specific protein, an enzyme specifically. And so what should be jogging your memory is, lecture, what are you thinking about proteins? What determines their function? What can prevent them from doing their function? And what may speed up the function? So in our case, we're going to see what things will make this vegetable, this potato, brown quicker or slower. So in our experiment, what you want to make sure is that you have your different groups. So we're going to have 3 different types of hypotheses that we're creating for 3 different groups. So it's almost like we're doing 3 different experiments. So the first experiment will be about salt. You'll be doing different salinities. So you have a 0, a 10, and a 25% salt. I'm not sure if these are the exact levels of salt that you have in your actual data. They might have changed because they change from semester to semester. Then you have a pH group. So you'll have different pH levels. In this picture we have 3, 8, and 11. So one thing that you want to keep in mind as you're looking at these different groups, what would your control be? If you're just doing the salt experiment, would 0% salt be your control, 10%, or 25%? Because you'll have to identify your controls and your experimental groups as well in your summary. And the same for the pH. What of the pH, 3, 8, or 11, would be considered a neutral zone or a control? The last one that we'll be using is temperature. So we know that temperature is a really big element in maintaining homeostasis for our own bodies. So that may give you a clue as to enzymes may need to have specific temperatures in order to do their functions. So if we expose them to hot or cold temperatures, then how might that affect the rate at which the enzyme is making the potato brown? Caution, do things safely in lab, even if you're online. Okay, so remember that whenever we do experiments, there's a lot of precautions that you need to take in order to make sure that your data doesn't get all mixed up. So even though you're not performing this part, it's a good reminder to label all of your tubes, not get them mixed up. And the same with your data as you're writing them down at home. Make sure and label everything so that you don't get your numbers confused. So after you've collected all of your tubes and your juices, so you're going to have tubes with salt, a bunch of tubes with different pHs, a bunch of different tubes that you will either put in cold or hot water. And again, you're not doing any of this at home. We'll do it for you and give you the numbers. But you want to make sure you understand all of your different groups. So after you've collected all of this information, after 10 minutes, you're going to add catechol to your tubes, to every single one of them. So we have different groups, but we want to treat them all the same. We're keeping that stuff constant about it. So whenever we add the catecholase, that will help us see the effects of the enzyme, or I'm sorry, the catechol will help us see the enzyme effects of catecholase. So when we're ready, we transfer it over to this cuvette. So this is the cuvette that I said we put all of our liquids inside of, and it goes inside of the actual colorimeter. So right now you can see this juice inside of the cuvette is somewhat opaque. It's pretty clear, not terribly brown at this point, but it's also not exactly translucent either. So that's essentially the effect we're looking for, is what color is it, and then also how opaque or translucent is it. So this is your colorimeter right here. You always need to calibrate it before using it, and that's what this slide talks about. And this second picture over on the right is what shows, it shows us how we put the cuvette inside of the colorimeter. So if you were doing this in person, we would do it in Logger Pro, very similar that we did the ice and ice water experiment with. But we're just going to use it for its reading. So this reading that we're looking for is the transmittance. So you can round up just to 1 decimal. You don't have to do a lot of decimals, but this is what this reading will look like. So this object has 38.7% transmittance. That means light only shines through about 38% of it. So you would imagine that this object is more or less transparent. If it's less transparent, it means not much light is shining through, a bit less than half. So you would assume this potato juice got about 62% brown. So how did I get 62%? So, this is an important slide. I forgot to tell you guys that. 100% transmittance is how we start out, right? So, if we subtract the 38% that we are left with on our reading, then we assume about 62% of the browning still occurred. So you are welcome to use either the 38% or the 62% as your number on your graph. Either works, but this is where ravioli comes in. What's really important to remember is that you want to describe it correctly. You want to make sure that you know what that 38% means. So if your graph is how much transmittance occurs, then you'll want to use the transmittance numbers, that 38%. So you want to make sure that you know that it's still turning brown. Okay? You could also do the final number, which is 62% on your graph. Just make sure that your graph says how much light didn't shine through. It's up to you how you display it. and we'll have many different looking graphs this week as submissions from students, but there's lots of different ways to be correct. If you have concerns about how to display this value, which many of you probably will, feel free to reach out to me and I can kind of describe it for you a little bit better. So don't forget to clean up after lab. Don't be that person that leaves all their stuff dirty. If you didn't do it last week in lab, just know there's always redemption. Next week you can clean up. More cleaning up, just read all this stuff and know that we value cleaning. Okay, so hypothesis. Remember that your hypothesis needs a causal explanation. We talked a lot about that last week in lab. So the goal of a hypothesis is to be testable, though. It doesn't have to be supported. You don't have to test that you think that pH will make browning occur faster, or enzyme rate will occur faster, or something like that, because certain pHs do that. And then if it doesn't end up supporting your hypothesis, then you might think, oh, I need to start over. I don't care if you support or unsupport your hypothesis, because remember, we can't prove or disprove them. But I don't care that you necessarily have data that supports what you hypothesize. I just want you to talk about it. If it doesn't support your hypothesis, then just talk about why it turns out it doesn't support it. You might find with some further Googling that it turns out you were wrong, and that's okay. So an example hypothesis would be enzymes exposed to pH 11 have lower activity because bases denature enzymes. So denaturing is a big theme in this week's lab, so you may want to go in that direction with some of your hypotheses. But remember that you need to create 3 hypotheses, one for each of the 3 conditions. So one for pH, one for salt, and one for temperature. So these all 3 will be included in your introduction. You don't need to create 3 different lab summaries or anything like that, but you do need to have 3 different graphs, one for each condition that you are doing. The other activity that we'll be doing is citing scholarly literature. So we did a lot of work last week with citations and library resources, so we want you to be able to use them now. So don't forget these tips for paraphrasing scholarly literature. So is paraphrasing the same as quoting? No, do not quote. What you're going to do is summarize your content into one sentence that you've created. Do not borrow phrases. Don't even borrow the way that they say things because you think it's nice and maybe more scientific than them, because those are their phrases. That's their intellectual property. You usually want to write down what you think it means, and usually I'll walk away for a few hours, come back, and then rewrite it again. That way it's pretty far away from the original quote, but still contains the essence of the quote. So sometimes you have to paraphrase an entire paragraph into one sentence, and that's similar to what you'll be doing here. After the paraphrase, you put the name of the author, comma, and the year. So we're going to have you do a scholarly citing activity. So at the end of your sentence, or at the end of your lab summary, we want you to summarize this into one sentence. So you'll read through this entire paragraph and then give the general gist of it, the Cliff Notes version, in one sentence with the proper citation at the end of the sentence, and do not use quotes. So for the article information, you'll need citation information. Here's the information from the article of the paraphrase that we just gave you. So you'll need to have a literature cited part at the end of your summary that also includes this. So just to review, the How to Write a Lab Summary document is in your resources module in Canvas, and I believe there might be an example lab summary up there for you as well that should help. The lab summary should be uploaded to Canvas by Friday at 5 p.m. You don't have to have it due at the end of your lab, obviously. Since it's a snow day, we're just going to make it the same as all other online activities that we do. And then also make a Flipgrid video about this week's lab successes and respond to the prompts that you are given in your Flipgrid discussion. If you have any questions about this week's lab or about completing it, feel free to email me. I'll email you back, or we can set up some Zoom time together or office hours in person. I know creating a graph is probably something that you guys feel, some of you probably feel like this is still new to you. Feel free to attempt and then reach out to me if you need help. And if you need help for just general studying in lab or lecture, feel free to reach out to me as well. I'm here, but know that after about Friday at 3:30 p.m., there's very little I can do for you in assistance. So make sure and get these done even though you have today off. Don't procrastinate like me and Ravioli might be tempted to do. So stay safe and warm, and I will see you guys in 2 weeks.